Good morning, everybody. My name is Ed Sitar. I'm the Chief Financial Officer of Cancer Genetics, and it's my pleasure to welcome you to our Portfolio Day. We have an exciting group of presentations we think you'll find informative. Agendas are in your package. The times are approximate, and we're going to do our best to stick to the schedule, but please bear that in mind. We're going to ask that you hold your questions until a speaker is finished. So the questions after each speaker, but please hold them to the end. And it is my pleasure to introduce our CEO, Panna Sharma.
Thank you, Ed. Thank you guys for all attending. This is going to be quite an informative day. We're going to open up the day with Dr. Farrugia from the Mayo Clinic. He'll be dialing in at 10:30 to give an update on OncoSpire. He'll be followed with Jane Houldsworth. Jane, you want to raise your hand just so everyone? This is Jane Houldsworth. She's our head of R&D. She'll be going and diving into programs you guys are probably already familiar with, which is our lead franchise in hematologic malignancies. A lot of progress has been made there. And also we have Dr. Gowrishankar. You want to introduce yourself? She'll be reviewing our renal panel. We think that is going to be an absolute blockbuster going forward. We'll talk about what's very unique in renal. And we have Marie Samulis to give a perspective on what we're doing to commercialize FHACT, which is in market and growing rapidly. So we've got a lot of great content to review today. Before we get started, I want to give a little perspective on how we view the market overall and what is unique about cancer genetics. I think we all know that cancer is an increasingly global phenomenon, not only for patients, but really it represents over 40% of the class of trials and nearly 50 to 60% of the spend in healthcare and disease maintenance and diagnosis. We're well positioned today, post the acquisitions, in not only the United States, but also in India and China. The way we view the markets is that there's two very clear and distinct segments. There's the clinical market, where we're serving cancer centers, patients, and really providing insight to help manage, treat, and in many cases, provide therapeutic guidance for cancer patients. For the cancers that we serve, there's about $6 billion spent in the United States, roughly $1 billion in India today and growing rapidly. In China, we are not serving the inpatient or clinical market, and that's not part of our strategy. The other segment that we're very excited about that we think is a major differentiator and is important is the biopharma and clinical trial segment. There are a number of reasons that is important. Number one, there's more and more compression behind an active and more importantly a successful trial, and how those tests and methodologies are then implemented into the clinical setting. Also, it's very important that more and more patients are now part of clinical trials, and that's a way to differentiate. Less than 5 years ago, the FDA indicated that it wanted patients, especially in oncology, to be part of clinical trials. At the time, the number of patients participating in clinical oncology trials was less than 6%. That number is now grown to over 15%. And in some centers, such as Dana-Farber or others that you go to, the number is even higher. So being able to participate with thought leaders in the clinical community is very important. That market, in the areas that we serve, is about $2.5 billion in the U.S., about $400 million and growing rapidly in India, and about $1 billion in China. And those are markets that we're servicing today as well. So what are we focused on? What are cancers that we're going to talk about that we're uniquely positioned? Our strategy is we cannot serve every cancer, but we do go deep in very specific conditions. Leukemia, lymphoma, cervical cancer, renal cancer, and multiple myeloma. So just in our areas of focus today, there are about 1.7 million new cases, and because of protocols, because of the internet, because of diagnostic technologies, there's becoming a flatness. There's no longer this delay between new methodologies that are developed in the US or Europe and what doctors want in Brazil or India or China. It's the same thing that happened with mobile phones and with technology and with everything else. If you get diagnosed with leukemia today in a major metro center in Shanghai or in Hyderabad or in New York, chances are that the leading clinicians all want to do the same type of panels. So we're very uniquely positioned to have global standards to accomplish that. It makes us very attractive. One of the most fundamental things that we focus in on is we're very uniquely positioned, is the backbone of what we do has to be validated by leading research centers. So for a company our size, we have research and collaborations and validations and relationships with centers such as Sloan Kettering, Columbia, Cleveland Clinic, Dana-Farber, Memorial Sloan Kettering, University of Iowa, Chaminade. So we have a tremendous list, as you can see behind me. It's an area that we spend a lot of time on for two big important reasons. And again, these are differentiators that I think need to be appreciated better by both our investors and also by the analysts. Number one, long term, it's very important to gain insight, innovation, and access to these ideas. Number two, and this is equally as important, it's very important that we can validate with these thought leaders. So as we do these trials, such as with Beth Israel and the University of USC at Keck, they give us great data to then go out and get better economics, get better validation, and more importantly, publications, and then those translate into better reimbursement by insurance companies. So how are we focused? If you look at where we are generating revenue now post the acquisitions, our proprietary programs drive value really from what we call from bench to bedside. And that's very important because we're uniquely positioned to not only help discovery efforts, but also help efforts with the biopharma community with their trials, and then most importantly, drive that also into the clinical community with patients. And so you see some of the companies and groups that are representative for that type of work. And so, as we create this company, our goal is to create a unique company that has the ability to be the oncology partner from bench to bedside globally. So how are we developing that footprint? You guys will, at the end of the day today, you'll see, we'll give you a tour of the facility here in RTP of the Gentris acquisition. You'll kind of understand and appreciate why we acquired Gentris. But now we have in the U.S. facilities in New Jersey and North Carolina, and we also have now facilities in China and India. So if you're a biotech or if you're a major research institution or a cancer center, having global harmonized standards across these sites is very important. We're now about 170 people, all very focused on the personalization of medicine. So it gives us a footprint that, unlike many other emerging companies. So what are our drivers for our proprietary programs? We're going to spend a lot of time today going deep into our microarrays, into our next-gen sequencing panels, and really into understanding how we approach developing unique content. So let me walk you through what the hallmarks are. Number one, we look at, is there a clear and unmet patient need? So it's unlikely that we're going to launch and invest in our dollars and in the investors' dollars in programs that are me too. So we really try to focus first and foremost, if we're going to have unique R&D efforts, is there a clear and unmet patient need? Does the clinical community, do our academic and research collaborators, we validate it with them, we validate it with our SAB and our MAB. Second, is it something that we can develop IP around? Is it something that's unique? It's very important because we don't want to spend our investor dollars on areas that are not going to be defensible long term. And I know that's an area for a lot of people to look at and say, is really anything in the genomic space defensible? Great question. I think part one answer is, it's clearly you can't not patent genes, but you can patent algorithms. You can patent certain processes that are not readily observable, and that's what we focus in on. Not the level 1 type of analytic, but really level 2 and 3. What kind of correlations can we make across genomic data or to specific disease end states? And we think that long-term, that IP position will be very important. Number 3, can we leverage the existing or the routine sample taken in patient care? It's an important point, and if you look at our tests, whether they be in kidney, which Banu will go through, or in cervical, which Anna Marie will go through, we always try to design our tests by taking the same existing sample that's taken today. It's oftentimes a point that's lost because there are definitely great technology out there and tests out there, but they require either a fresh sample or additional material or 2 types of samples, and those I think are going to be problematic. So we always look at what is being taken today from the patient. Doctors have told me, and just doing my own research, if I'm not paid to take out an extra sample, I won't. If someone's there who's not going to be able to do this for this test, I'm not going to take it out. You work with what I have. Oftentimes it's formalin fixed. Oftentimes it's material that they're already taking samples for. And so part of our strategy is we work with the existing sample. In cervical, we'll go through that. We work with the existing pap smear or the HPV specimen that's already fixed. In renal cancer, we work with the needle core biopsy that's already taken. So it's a very important point. Next one, as we develop our proprietary programs, can we validate it? Do we have a partner out there who's willing to take our technology, our test, our insight, and give us their precious specimens? There is a, it's a good point for analysts to know, but human specimens in the United States that are clinically annotated and come with the information, the price for those specimens is skyrocketed. And it's not just because the pharma community wants them, but also now these large-scale translational efforts across the country have driven the prices up for samples. People are very, very reluctant to use them. They give them up. They're harder to get approval for, and it's a differentiator for us having access not only to samples, but having research collaborations where we get the clinical insight and where they're willing to invest their precious sample, their precious clinical data, and their medical records to help us develop and validate new technology. Again, it's a major differentiator, and we always try to figure out who can we validate with. And it tells us if they're willing to validate, if they're willing to spend time and energy, that there's something that's there, it's important. I would always be reluctant of any company that tells you that, hey, in our lab we did X, but no one else is validated. I think it's a major red flag. So our continuing philosophy is not only develop unique insights, but make sure you have tier one partners out there that are going to allow you to use their library and spend their time on validating it. Next, and very important, I think a lot of this we've talked about before, the medical world changes slowly. Clinical care changes probably even more slowly. It's why today we continue to do things such as white blood cell counts, we continue to do morphology and staining. These technologies have not disappeared because NGS exists. In fact, they've become additive. Our strategy, and we think long-term it's very important for our investor community to understand, is that we offer a complete program. So when we get a sample in our lab, we not only can do the cutting-edge genomic work that people expect us to, but we can also do the routine work. And of course, all of our routine work is very focused on complete programs around that disease state, such as complete CLL, complete renal, complete cervical, et cetera. That's important for two reasons. One is that it avoids splitting the specimen, and number two, it allows us to gather and corroborate additional omic data or additional biological data to constantly improve the offering. So those are the kind of the fundamentals of kind of our philosophy and the drivers for our proprietary programs. So with that, I'm going to open it up for some Q&A before we launch into OncoSpire. In India and China, those are two very different sites, so let me give you some color on that. In India, we have 33 people, 6 salespeople, and that is already an NABL accredited lab, which is the equivalent of CLIA here in the US. In fact, we will go for CLIA in India as well. We expect that this year we'll probably have an incremental 100 to 200,000 in kind of IT robustness in India, and next year we expect somewhere in the range of 300,000 to 500,000. The reason that number is so low is that we already had pretty significant investment because it was a 14,000 square foot NABL accredited lab with NGS capabilities with existing customers. So a lot of the investment up front had already been put in, so we were fortunate on that part. We do think that the bigger investment is going to be people and process rather than equipment in India. In China, it's a very different situation. We have 3 people. We just, in fact, last week reported out and received our very first samples, so we'll be talking about that. We'll be issuing some releases on that shortly. So we are in market now in China, and again, we're able to leverage the investment that the prior Gentris folks put in over the last 2 years. They invested somewhere in the range of $2+ million getting that facility up and running. And it's a pure genomic and repository facility. Again, the focus of that facility is to serve the global biotech and pharma companies. We think near term this year, we're still assessing it, but we will not be making major investment this year, but we expect next year to be having to spend somewhere in the range of $1 million over the next 2 years. Yeah, absolutely. So this is a question from Brooks talking about the ramp of the business. Let me start with the China business first. China, we're really serving global biotechs and we're trying to piggyback on the existing relationships. The reason in China and India both, you cannot ship human specimen or samples outside of those countries. So you really can't send them to Europe or US for analysis. You can under certain conditions, but it's very challenging. So pharmas, as they do trials in places where the dollar goes further, they have to have the analysis done there. China's operation is very, very focused on serving the global biotech and pharma and piggybacking on the existing relationships that we have, like with Roche or Novartis or Merck or J&J. So we think that'll grow pretty much alongside the growth of the RTP facility. Historically, they were not servicing them, and again, we just started last week or last 2 weeks. Is that right, Michael? Yeah, so it's very near term, so we expect a lot of growth out of China. In India, they are serving the local clinical and research market, so it's a different phenomena, and we expect, readily, 50 to 100% type growth in the India market near term. A lot of that growth, what we're seeing in the pipeline, is NGS focused, so our rate limiting factor will be NGS and bioinformatics, and we are going to have to be more disciplined by the type of business that we take in India. So that's a good quality problem to have. So we're going to focus more on clinical, more on high-end NGS and research, and again, since we've acquired it 2 months ago, we've already, we'll be making an announcement again, we've already closed a major cancer deal with a hospital there as well. Brooks asked a question about the reimbursement environment in India. So in India, what we have is we have a country where cancer is rising rapidly. They expect it to increase 5x over the next decade. A lot of that is due to not only that we have improving diagnostic tools, but really because the westernization of diet, lifestyle, et cetera. And so as this increases in India, what we see is we are not well equipped today to serve the billion-plus people. What we're well equipped to serve is really the metro markets and probably the middle class or the affluent middle class. And if you narrow that down, our real total addressable market size in India for us is about 200 million people. And people talk about the middle class there being about 350 million, and so we take even a subset of that. Now, obviously, our goal and our ambition is to be able to expand that, but the reality is that the real market is about 200 million for us. And there, they do have both nationalized insurance, but they supplement it with their own private insurance, just like we do here. And out of that 200 million, there's 100 million that have tremendous purchasing power parity. So they're willing to spend $300, $400, $500, $600 on their own tests. And so there's a lot of individual and consumer control. It's a very different dynamic than the US market. We're here, sometimes people complain about having to spend $200 on an x-ray. There it's not uncommon for people to spend $100, $200, or $20 out of their own pocket. Different cultural dynamic. So the reimbursement is not going to be the bottleneck for us.
Could you just talk a little bit about the pricing of some of the more basic testing in China and India and around the world relative to the United States, and then some of the next generation molecular testing you're doing and how it compares, and then maybe your profitability and your ability to drive profits in those various geographies with your cost structure in different geographies. I am just trying to understand how much scale you need, how much growth you need in India and China and around the world to be able to drive profitable operations in those countries.
Great question. So I'm going to take China a little bit off the table. That's a great question, because in China, again, Charlie, talking about the pricing and market dynamics, we are serving global biotech and pharma. So we're not really focused on the Chinese reimbursement market lens. So we're really set up there to be an arm of our global trials. So we price the same as we price here. So it's not, that to us is not a challenge. Same in India for pricing for trials. We price as we price here. We might price a little lower because of labor arbitrage, but that's not the significant thing for us in China. In India, where we are serving the local market, yes, there the pricing is different. And the interesting thing for our COGS in India versus here our COGS is 60% plus. Ed, is that right? Labor. Rule of thumb, 60% of our COGS is labor. Depends on which category. The rest is everything else. In India, it's almost the reverse. 60% is everything else, 60, 70%, 30% is labor. So we think we have good control in terms of scale in India. However, the rule of thumb that I use is that typical reimbursement, end market reimbursement, is about 1/3 to 1/2 of what it is here. In the NGS, not the same because it's a newer technology, so your reimbursement there is probably 20 to 30% lower. Not necessarily half, but for routine, it's about 1/2. Okay, good. So we're going to take a quick pause, and then we're going to continue with going into OncoSpire, so some background. And we'll get Dr. Gianrico Farrugia from the Mayo Clinic on our line.
Hello. So we're going to go through this at a fairly rapid pace to allow questions. And what we'd like to give you is an update on where OncoSpire has come, given that quite a lot of progress has been made in the last couple of months. So I know a lot of you know this, but as a very brief background, we decided to create this collaboration with CGI because we strongly felt the time was right to use next generation sequencing, and you see that, of course, and I know most of you know this, as NGS on all the slides, to transform healthcare. And so what we did is we went through our 50,000 employees and asked for what are their best ideas, got back many, many applications, and then chose these 3 that we're going to be talking about. And so we formed these 3 groups that have come together and are really focused on what we think is the most important thing, which is how do we leverage next-generation sequencing, how do we leverage CGI's resources, Mayo resources, to make an impact for patients by having new tests on the market. So, Panna, perhaps you can go to the next slide, which says multiple myeloma.
Yep, I'm there.
All right, and then the next slide. So I'm going to start with multiple myeloma. Multiple myeloma is a disease that it affects blood cells. And really our strategy there has been to develop 2 next-generation sequencing-based tests. Why? Because we feel that there now is enough sequencing done that we have a fairly good idea of literally about 99% of the genetic abnormalities that occur in multiple myeloma. And therefore it's time to take that information and make it into an easy-to-use test that replaces quite a lot of expensive testing that is currently done in a very uneven fashion. And that's important because it helps guide therapy. And while multiple myeloma was always seen as an early and lethal disease, we now know that there are many things that can be done that can markedly alter the course of the disease. The second would then is to tackle this really large group of patients who have what's known as MAGAs. And MAGAs is when there's too much of a particular protein in the blood, and as we grow older, a larger segment of the population ends up with having this MAGAs. And the problem is that we know that a small percentage of those people will eventually end up having multiple myeloma. Most others don't, and there is no current good way of predicting who is going to get it. And if you do, 1 is you alleviate a lot of anxiety, and 2, you of course have an ability to intervene earlier. So if we go to the next slide, please. We have already selected which genes need to be on the panel. There are 77 genes that we've selected to be on this panel that would tell, that will report on all the abnormalities that you see in multiple myeloma. And so now what we're doing is we've just sequenced the first 60 samples and are comparing from a very, how are we going to get this into widespread use, which is the best platform to do it, that is reliable, that is reproducible, and that is also economically feasible. This is something that we were lucky enough to have what you really would consider to be world leaders involved in this project. They're people who they're coming along from a long history of research in multiple myeloma. They're sort of the 3rd generation investigators. They've already published 3 papers on this. They're going to be having a presentation at ASH that's coming up as the largest meeting in the space. And they've already figured out that this works best if we collaborate internationally and have established that, including with a group in Germany. Next slide. So that's the multiple myeloma one. It's probably the most advanced. It's the one that is now already approaching what's the platform and how do we get that platform and make it really work at an international and national scale. Panna, do you want me to take questions on each one of these separately or at the end?
We'll take questions at the end.
All right. So we're now going to switch gears and move to the second one, which is follicular lymphoma. So lymphoma, as again, you know, is another disease, affects a different cell type, a different blood cell. Now, follicular lymphoma is a very interesting lymphoma in that it is very common. It is the most common of what's known as the slow lymphomas that grow slowly. But there's something that is a characteristic of follicular lymphoma that is very hard to deal with at the moment, which is why most patients have this very slow growing, what we call indolent course. A subset of patients that is quite significant will all of a sudden transform to a much more aggressive lymphoma, which is much harder to treat. And therefore, there is this huge need in the market to be able to tell patients their follicular lymphoma is going to behave and going to remain behaving relatively benignly versus the people that it's not going to behave benignly. At some point, it's going to switch, that we need to be so much more aggressive in therapy because at the moment, we have to treat everybody conservatively because we think most people will go slowly. But when we do that, then we put at a disadvantage the people that are going to have this much more aggressive form. So if you go to the next slide, please.
Okay.
So the focus here initially is to develop a next-generation sequencing-based test against a particular gene, BCL2, to help us make the decision that I'm talking to you about. And so we think that there is a significant market for this, and that this panel, starting first focusing on BCL2 and then expanding, will really become a standard of care because there isn't a standard of care at the moment. There is this sort of big unmet need that we think we have a very good opportunity to become market leaders in. So where are we? We've actually secured all the samples we needed because, of course, one advantage we have that you know is the size of the patient population at Mayo. One of the issues is it's easy to do this on big samples, but you've got to be able to do it in a reproducible way on very fine samples. Sometimes they're called aspirates, where you just have to pull back on a needle and you get a sample, or when you do some other interventions and you get small samples. And so we're now moved and are working on these smaller samples, and our first job would be to get this first BCL2 to market, to be closely followed then by version 2, and version 2 will be expanding on what is really known about some of the other genes and how they prevent our body from developing antibodies that can prevent the progression of the disease, and understanding those antibodies, something called AID genes, then you can develop a second generation. The last thing I want to talk about, Panna, next slide please, is lung cancer. So we're switching gears a little. The first two were blood. Now we're talking about lung. And of course, lung, despite the change in smoking, remains #1 in cancers. And so there are many, because it's such a broad area, there are many areas to focus on. You identify the people who are at the greatest risk of progressing. You identify the people who have the disease, what kind of treatment to do, and you identify how to select the treatment. The first approach we are taking, next slide please, Panna, is actually dealing with what most people don't understand is that is a significant problem for lung pulmonologists, for oncologists, and for surgeons, which is very often somebody who has lung cancer has in their lungs many nodules. And these nodules, as we grow older, a lot of us have them, but people who are going to get lung cancer happen to have more of them. And some of these nodules are benign, and some of them are malignant. But even within the malignant, it's very hard to tell if it's the same tumor that has gone to a different place, so that's a distant spread, a metastasis, or something where two tumors, because of the background being conducive to getting cancer, two tumors have developed independently. That is, you'd consider that to be a nuance, but actually it is fundamental. Because in one case, you're talking about a disease that has spread. In other case, you're talking about, no, it's two cancers that have happened, and you can treat them as if the both were localized. Big difference, surgical approach, big difference in therapy. So, again, the issue is then, how do you work on very, very small samples, samples you get from an endoscopy and washing the lung to get cells out of? How do you work from very small paraffin-embedded specimens? How do you do it from cytology? And so what we're doing at the moment is taking initial samples where we have normal lung, we have lung that we know is a metastasis, and lung where we know it's not a metastasis. And you can see how this doesn't necessarily have to be only in the lung. The second lesion could be somewhere else, such as in the brain. And so our first focus is to develop a test that can guide physicians, surgeons, oncologists to make that really essential first bifurcation in their decision. 3 is, what are you dealing with? Are you dealing with two things that are the same or two things that are different? And so with that, I'll stop. Panna, is there anything you want to add from your side?
No, I think that's a good overview.
So Scott and I are here, and Panna's here to take any questions.
Thank you, Gianrico. For the first question from Sung Yi at Cantor.
Dr. Farrugia, was wondering, I have a couple of questions actually. Starting with the lung program, obviously there are a lot of other lung cancer tests out there looking at kind of risk of progression and things like, or aggressiveness of cancer, I would say as well, and was wondering how is your test differentiated, or could you maybe talk about how, looking ahead, how it might be differentiated?
And I apologize, I couldn't hear that. Panna, if you heard it, could you repeat the question for me?
Yes, absolutely. So Sung Yi was asking how this NGS panel for lung will differentiate from the other tests that are out there, NGS tests that look at progression and also look at some of the driver mutations.
That's a great question because the field is so big that, of course, there are many different ways of approaching this. Our approach is, at the moment, initially not to go after the progression, but rather to go after this very specific question that nobody else is answering. And the reason they're not answering is you cannot make the decision about two samples being the same or different based on normal NGS-based technology. You need a new way of identifying the sort of the origin of the tumor, which is very different from the genetic abnormalities currently in the tumor. And that requires a bioinformatic approach that is fundamentally different from other NGS-based approaches.
Okay, and then my second question is for the multiple myeloma. You talked about, I think, 77 genes. Will there be further winnowing down, or is that pretty much, do you think, the final set? And just kind of curious as to, not a scientist by training, so multiple myeloma, 77 genes, and follicular lymphoma, it seems like fewer genes there. And so from that standpoint, do you think that, is it just the biology of the cancers, I think, dictating kind of the number of genes, or I'm just kind of curious as to what, the 77 seems like a big panel,
Anna,
even
could you
compared
do the same
to...
favor for me?
The 77 seems like a big panel compared to what's out there for other panel-based testing as well, right?
So Sugi's question is, basically, how are we arriving at the size of these panels? Do we feel like they're small, large? She thinks the 77 gene panel for multiple myeloma seems large. Is that correct? From her viewpoint? She says compared to other panel-based tests that she's seen. And what was the second part? Is that it?
Follicular was small.
And the follicular seems small, which, BCL2 is a large gene, though. And it's deep. Yeah, so she's trying to understand what, how are we making the decisions on the genes? Is it biology? Is it literature? Is it, she's trying to understand the size of these panels, how we arrive at them.
Thank you. So the first is, both answers actually are the same. And because we had such a rich sort of pool to choose from, we went after really international leaders. that have, it's not like they're entering the field. They've been doing this. They have their own academic careers in this area they've contributed substantially. For example, for the multiple myeloma, I think the Mayo Clinic had, and I'm not sure this is quite right, but had not the vast majority of papers ever published in the space, in the multiple myeloma space. So they were able to go and look at their own data and other people's data and figure out how many genes are there that would capture the vast majority of useful information without doing too much. We originally said we could do it with 47. And then as we looked a little further, started to share samples, we realized that 77 is probably a more accurate number to get to where we need to. Will that number ever change? Yes, it will. The nice thing about next generation sequencing and how you build these gene panels, that there is some flexibility once you decide on the platform of which genes to leave in and out as more information comes out. So at the moment, we do feel that 77 is the right number.
Let me add some color.
The second question.
Yeah, I'm going to add some color commentary, Gianrico, if you don't mind. So if you look at,
yeah, of course.
If you look at the panel, there's another panel that's out there offered for heme, the Foundation Heme panel. That's about 400 genes. And if you compare our very focused multiple myeloma panel, which again answers questions that they can't answer, which is replacing not only the existing technology, but then also the predicting change, which is a major market. So predicting MGUS to MM transformation. We're only looking at 77. If you compare the 77 gene set and how we're looking at it versus the Foundation's 400, roughly, it's about 50% of those 77 are even covered on that 400 gene panel. So this is really novel insight that's not available. And that's, again, we're going back to our, this first slide, we're looking at not only unmet needs that existing panels cannot do, which is predicting change and replacing existing testing, but then developing new, unique insights. And so it's really, the number of genes is not as important as what are the questions that we're going after. And again, even in lung, there's no test that will be able to give you the answer to the question about, is this multiple independent lesions or is it one metastatic and where is it going? There's no test. They'll predict the existing stuff, which is progression, is it small cell or non-small cell, and does it have BRAF mutation or not. Those are all, again, commonplace and they're well served. So we're trying to go after and advance the field. Again, sorry, it's a little bit of an advertisement, but then, it shows you why we're going after those topics. Go ahead on BCL2, please, Gianrico.
Yeah, except I'm going to use just a little prompt on BCL2. The question was?
Oh, why BCL2, or what? The question was, is that enough, or will it change?
Yeah. And so here, of course, Dr. Shiganti in CGI as a founder has a tremendous sort of leadership role in this disease, and we have our own information that strongly suggests to us that if you want, in this case, this is a critical decision point that the cell makes that then predicts where it's going to go. And so in this case, unlike the multiple myeloma, where you need 77 genes, here you have the opportunity to do a lot by initially going after BCL2, and at the moment, there isn't an easy way of doing it, and that's where we feel there's an unmet need, as well as we have the expertise to go after it. So that's why BCL2 in this one. This is one of the diseases where one gene has a more critical role than in multiple myeloma, where there are many genes that have less critical but combined greater roles.
This other point there also, BCL2 is also a very active target for a lot of these agents as well. So, Jane?
So follicular lymphoma at the genetic level is almost invariably defined by a rearrangement of that gene in a translocation, which is the 1418 translocation. In this particular case, it's mutation rather than translocation that the literature suggests and the studies have suggested may actually be involved in the transformation of that indolent disease into the more aggressive disease. And that's the power, and the reason it's this gene that's been suggested to do that. And that's why it's this gene.
And there are literally hundreds of different types of mutations in BCL2. So it's not just, is it mutated or not? Thank you. Oh, one more question? Sorry, one more question.
I guess in general, I was wondering if you could discuss how the cost of goods change when you have an increase in the number of genes that you're looking at in an NGS sample. Is it dramatically different or not? And what percentage of follicular patients progress to a more aggressive form of the disease? And what is the differentiating molecule or gene that you're looking at there?
So Gianrico, he's asking a couple of questions. Number one is the question of what number of follicular patients transform was the first question. And what was the... And then we'll cover COGS separately. So if you want to ask first about number of patients that transform in follicular and get some more common color on that.
So about 20 to 30% of that group of what's called non-Hodgkin lymphomas are follicular lymphomas, and out of those, about 30 to 40% will transform.
And how long does that take typically?
That varies quite widely, but sometimes it can take 15 years. Other times it can happen much earlier. Once it happens, you tend to have less than a year of survival. So you see how difficult this is for physicians at the moment and for patients, where we're telling everybody at the moment, look, you have 60% chance that you have a disease that is not going to progress. You're going to survive 12 to 15 years. You're going to be, and therefore we can manage you quite non-aggressively. But we know that a certain percentage, between 20, 30, up to 40%, you'll have something will dramatically change, and it will change in a very short period of time. And when it changes, then really your survival goes in a totally different direction. And we have to, and that's why we see this as not as a Me Too test. This is a transformational test that's why we feel that this will capture that market, because it is essential information for the physician and the patient.
So the follow-on question was how frequently will those people have to be tested? Every 3 months, 6 months, year? Is there any standard that exists today for the high-risk follicular?
So because we actually don't know, there's no test at the moment to predict progression. We don't know the answer to that question. We don't know if you do it once and that information is enough, and there's going to have to be research to figure out how often. It is possible that you'll have to repeat it, but there really is no information to tell at the moment how often you're going to have to do it.
I'll add some color on that. So even in the early data that we've seen historically and with some of the BCL2 mutations, we do see two very interesting sets of patients. The ones that transform quite rapidly in 3 years or so or less, and then we have another group that is out 7 or 10 years. And so that's kind of interesting. We're getting to see kind of two different types of signatures or sets of patients, ones that are kind of rapid transformation and one that kind of are the mid-range. And so it's, it could be, again, they'll have to be monitored, but without more trials, it's unclear whether you test every year or every 6 months, but a lot of that will depend on the clinician's choice.
I'm just curious, if we identify these two groups of patients, are there different treatments that can be brought to bear so that you hopefully change the outcome or improve the patient's condition or situation? And then the second question I had was if the doctor could just talk about some of the key milestones he's looking for over the next period of time and help us understand sort of the time frame he thinks it might take to get to those key milestones.
Great. So I think these are two different groups of questions. So let's take the first one, which is what are the actionable events related to this type of testing? Are there therapeutic choices or treatment decisions that it helps guide or make? And are they well known or are they emerging? So that's the first one with regards to, I guess, both follicular and multiple myeloma, right? Okay.
So the first one, the answer is yes. So we are blessed in a way that in hematological malignancies we have lots of choices, but chemotherapy, even targeted monoclonal therapy, has its disadvantages. And so you don't want to use a big stick when you can use a little stick. So if you know you have to use a big stick because it's going to transform, you'd be much more aggressive. And there are now there are not only established, but also emerging new therapies. So yes, there is a therapeutic reason for doing this. In fact, the same applies to MAGAS and lymphoma, MAGAS and multiple myeloma. It's not only are there things we could do now, but we know that the therapeutic pipeline is fairly rich.
Very rich, yeah. The second set of questions was really about what milestones do we anticipate hitting over the near term, midterm, long term? What are the milestones that we expect for these programs? And when will we hit them?
Yeah, I mean, roughly.
Yeah.
And I don't know, Panna, if you want to handle that one or not, but one of the things we early on decided to do, as well, of course, CGI has a lot of experience here. Us through the Center for Individualized Medicine, we've also been fairly active in sort of the business aspect of this. So every one of these has statements of work that the scientists have signed with deliverables, and we've asked them all to deliver with a time frame of 18 to 24 months, with milestones that Scott, correct me, but they're every 3 months,
Every 3 months, we track projects regularly. We formally meet as a scientific review group to review the results every 3 months. We meet face-to-face every 6 months to review the results. But the milestones are meant to put us into a clinical testing environment within 24 months. And the way I would stagger them is the lung cancer test at this point is closer to the 2-year mark, and the multiple myeloma test will be much closer to the 12 to 18 month mark.
And we're already, yeah, so the way we look at the milestones, we have some papers coming out for multiple myeloma. That's closest to market today. The panel is set. We're in 2 big collaborations with global centers, and we're very confident that post-ASH we'll be launching. We'll be optimizing for clinical launch next year. And we think we can replace a major percentage of the existing multiple myeloma testing. We anticipate it being available through Mayo and CGI, potentially others. The second one will be follicular. Again, we're very focused that panels, will migrate a little bit. And again, we think that'll be a late next year event. And then, as Scott mentioned, lung is more aspirational, still early, and we expect that to be a 16 type event. But again, that said, we're finding new things. We may launch something more targeted even in 15. But the access to samples and access to the research is pretty phenomenal with Mayo, and that's part of why we drove this. So we're, part of the surprise was that we did expect follicular to come out first when we first started this project, and we've been pleasantly surprised with how quickly we've been able to progress in multiple myeloma, which is, a little more challenging potentially in terms of the number of sites. But again, through the joint work, and as Farrugia mentioned, this is the 3rd generation of researchers at Mayo and multiple myeloma, so we're really tapping into a very rich heritage. Okay, good. I know we're running behind a few minutes, so we're going to go ahead. Gianrico, thank you for your time. Thank you, Scott. We're going to move on to the next chapter.
So it's my responsibility to take you and walk you through some of the proprietary programs and projects that we have at CGI and how that fits in with our testing portfolio. So I thought I would start off and take a step back. You've been thrown into the world of multiple myeloma and follicular lymphoma, but I thought I'd just give a very brief overview just to put it in a perspective for you, since one of our foci is hematologic cancers. So just for that reason, our blood cells start from a stem cell, which then has 2 major arms. You'll hear us talking about the myeloid arm and the lymphoid arm. In the case of the myeloid, we have disruptions of the maturation and differentiation of these cells. In the cases of the myelodysplastic diseases, it's resulting from a problem with differentiation of a particular cell type in the red box arm, so that we get fewer of those cells in the blood. In the myeloproliferative neoplasms, or MPNs, these are chronic diseases like CML, whereby you've had overgrowth or some abnormal growth of one of these cells, again in this red arm here, in the myeloid. As opposed to the acute diseases, such as you'll hear from AML, where you have a massive abnormal proliferative overgrowth aggressive disease versus chronic disease. On the right side here, for the lymphoid malignancies, we have lymphoproliferative disorders. And these can occur in the B and the T cells and the natural killer cells that are involved in the immune system. And also, they can physically occur and present in the bone marrow, the blood, the lymph nodes. And in particular, what we're going to talk about today are the ones that occur in the lymph nodes. Those are grouped together biologically because they've passed through special germinal centers in that lymph node. And they share genetic characteristics because of that. These take on the names leukemia, lymphoma, and myeloma. So you've already heard a talk about multiple myeloma, so it belongs to this side of the picture, the blue and the red. So what's important about these groups, and you'll see, is that they're actually not just one disease. It may be called MDS, but it involves many different subtypes. And that goes for all of these. It'll be called an MPN, but there are many different, and they're defined not only by their clinical features, but also their underlying genetic features. The same with the lymphoid neoplasms. So what we do at CGI is we offer complementary testing that's platform agnostic to help the clinicians not only in the diagnosis, but prognosis of these diseases based on their molecular signatures and incorporate that in our proprietary tests in order to improve that and increase the utility. But in order to understand that, where is the need for these particular diseases, you have to understand it in the context of the continuum of care of cancer. And in fact, these 5 boxes would apply to most of the cancers. Obviously, we're talking about screening belongs to some of the talk that Dr. Farrugia was talking about with the MGUS in the multiple myeloma, how we can identify those patients who are carrying some low levels of particular alterations may go on and form then fully blown myeloma. Diagnosis, we are blessed, as Dr. Farrugia said, in that for many years now, because of the accessibility of these diseases, diagnosis has in many ways, based on what the cells look like in the blood or the lymph node, it's these areas that we've seen a lot of progress in recent years with the development of targeted therapies and how can we project how patients are going to respond. So let's first of all talk about the myeloid diseases and what we do. In myeloid diseases, I've listed the ones here that I've already talked to you about. Let's talk about, first of all, about the myeloproliferative. The diagnosis of these diseases, which are the low-growing myeloid cells, have been revolutionized in recent years with the advent of molecular testing to look for specific genomic alterations. And when I say that, it's as recent as a publication that came out in January describing the CALR mutation detection in the subgroup of these that don't carry a JAK2 mutation. We're rapid in bringing those into our complete testing, but in that respect then, diagnosis is very important in this group. For MDS also, you may have a clonal population, and that's important to identify, some of which we haven't fully characterized them yet. So without a diagnosis in this case, the clinician is often difficult to know how to manage this patient then. Also important in this disease is to identify those patients that are going to go on and progress to form a very aggressive AML. AML, on the other hand, important to prognose and to try and select the appropriate treatment selection, given that, again, AML encompasses multiple different subtypes for which genetic alterations underlie their phenotype. So what we've done, and we've built complete programs for the myeloid program around to assist in these diseases. And what you'll see now, and we're in the final stages of validating, is a 54-gene panel for NGS-based that will clearly further assist in the diagnosis of these diseases here, MPN and MDS. We currently offer single gene mutation analyses of these, like JAK-R, CALR that I just mentioned to you, This will be a much broader panel that will clearly allow identification of clonal populations of cells. It's also going to have these genes on it that are involved in progression to AML, that have been suggested to be involved. It also contains on it genes for AML that are involved with prognostic value. We're in the final stages of validation of that right now. The other thing that I wanted to bring up, and you'll hear about this in the news in the next few months, is that we're entering into a new research program into this myeloid area, and this will be in collaboration with some leading MDS, in particular MDS, clinicians and thought leaders. But I'm not going to talk further about that today. What I am going to spend most of my time on is the work that we've been doing in the lymphoid neoplasms. In these particular diseases, you've already heard about one, the follicular lymphoma, the so-called slow-growing or indolent disease that has the potential to transform into the more aggressive. So in this disease, the clinicians are more concerned about how am I going to manage this patient rather than just how do I treat them today. This is somewhat comparable for CLL. Again, it's an indolent disease, but in this case you have two main groups, an indolent and an aggressive, and it's important to distinguish the two. DLBCL, on the other hand, is in a highly aggressive disease, requires treatment almost immediately, and there the issues are different in the continuum of care. How well will this patient respond? I've listed here our proprietary product, MATBA, which is an array-based platform, which was designed to assess genomic copy number alterations at 80 genomic sites, for which we have patent protection and has been incorporated into clinical trials as biomarker assessment for various types of these mature B-cell neoplasms. It was designed in order to encompass these mature B-cell neoplasms as well as mantle cell lymphoma and multiple myeloma. I'd like to first of all address chronic lymphocytic leukemia, or CLL. So in CLL, there's about 15,000 new cases diagnosed each year, a number of deaths as listed here. What's an important feature here are two things. One, the median age of diagnosis of these patients is older, so it's more of an elderly associated disease. But what's more important is that the median overall survival is 9 years. So some of these patients can go for many years without requiring treatment, and some of them cannot. Very important upfront, when a CLL patient is diagnosed, what type of patient is this? Is this somebody who I need to treat now, or is this somebody who can go for 9 years or more without requiring treatment? So in that scenario, then, we have 7 to 8 times this number of patients who are actually living with CLL and who require treatment monitoring and to look at the genomic aberrations and other alterations that are occurring. This has become very obvious to us during the course of studies where we've done serial examination of the alterations that you see and how they wax and wane with time and in response to treatment, and after treatment, what clones are reappearing, what alterations do they have. They represent about 40% of all leukemias of individuals over 65, and as I mentioned before, there's about 2 equal groups, those who are indolent, those who are aggressive. So we have several initiatives then to address the clinical challenges in CLL. The first one is risk stratification, indolent, aggressive. How will the clinician know when to introduce therapy versus those who can just undergo active surveillance, and also then those who are most likely to have an overall poor outcome? But given the fact that over in the last several years there's been great progress and recent FDA approval of some of the new targeted therapies in these diseases, we have to take that into account as to how these biomarkers are going to fit in that risk stratification process with these new therapies. And that's a new clinical challenge, not only for us as diagnosticians, but also the clinicians. And so I'm going to address these topics. So we offer the only comprehensive complete risk stratification toolkit for CLL. It's platform agnostic. It measures a variety of abnormalities, the IGHV mutation status. We use FISH to look at gain and loss mostly at 4 sites. We use flow cytometry for surrogates of the IGHV. We have a limited low sensitivity mutation analysis, and our MATBA proprietary test, which I will talk more about, and the value that it brings to the complete. And now we're bringing in, and we're in the final stages of validation, ready for expected launch in the close of 2014, a highly targeted NGS panel for CLL. We have patent protection for MATBA, and most of these studies and the bringing on of this have been done in collaboration with leading thought leaders in CLL at Long Island Jewish, Dr. Conti Rai. So the value of MATBA, if currently FISH is being used by most laboratories to look for the gain and loss of 4 loci to risk stratify patients. It's only identifying about 15% of patients, but the remainder of the patients don't really have any distinguishment between are you intermediate, are you good, because I told you these patients are going to live for many years, a lot of them. In our research studies, using well over 200 specimens in our discovery set, we found that we could actually now look at 20 sites by MATBA, and this has been implemented clinically, 9 of which now are marked in red here associated with poor outcome univariately, and the others are now used to distinguish between the 2 groups into somebody who has an intermediate and a good outcome. It's these patients who have the ability to not have to be treated straight away. This will, and I think that that's obvious when you look at the next slide. And I apologize for all the Kaplan-Meier curves here, but I think it proves a point. So in our studies that we published last year, you can see here now, and I point that out, this is important, the time to first treatment as to when a patient will need to be treated. You can start to distinguish out the patient significantly, and I also draw your attention to the timeline of these curves, in that this is 10 years here. So we're looking at very long follow-up on these patients. Overall survival, we have some patients up here in this category who actually don't end up passing away from the disease for up to 10 years. That's very valuable information for the clinician. We validated it in different data sets from different institutions and collaborators, from Dana-Farber and Hackensack with our respective colleagues. In data sets, there were over 200 specimens. And what became obvious to us was that across institutions, they actually treat their patients in slightly different ways, and their clinicians are actually not guided as well as they should be as to when they can treat. So what we feel is important is that adding MATBA in now will help separate some out of these curves to find out when the time they're being treated. Also, and I think what's important is in this day and age of the increasing use of the targeted therapies, it's this feature, the time to first treatment, that's going to be very important to when the clinician is going to select to actually treat the patient. What we'd like to do, though, is to be able to build an integrated risk model. So and assign a risk for the patient. So right now, in our CLL Complete, we give out different pieces of clinical information. It's integrated into a report. But we'd like to be able to assign a risk, an overall risk that combines all the data together. And in doing that, and through that thought process, we realized that we had to have a very targeted panel in order to assess mutation analysis at a very high level sensitivity for CLL. And this is the panel that we've developed. CLL, I might add, compared to many other cancers like lung cancer and even the other follicular or diffuse large B-cell, has a relatively quiet genome. So it doesn't have as many mutations as many others. So we have our panel This is extremely challenging gene because of the size of it that we've had to encompass. TP53, the entire gene, BERK3, is highly prognostic of poor outcome. Our other two down here that we already do partial ones of, but we're an imminent release of this panel now. Armed with these now, we'll be working towards the risk model, which will be in order to provide that information on our CLL reports. How does this fit in now into this new era of these targeted therapies, in particular the B-cell receptor pathways that actually within the past year now have been approved for use in CLL? I've mentioned two here, the two that are involved in the B-cell receptor signaling pathway, the idelalisib, which targets PI3 kinase delta, and the ibrutinib, which targets the BTK. They target two slightly different arms of the B-cell signaling receptor pathway. So in both cases, they've been approved by the FDA for different indications, both of them to some extent in the relapse setting. The other one also ibrutinib for patients who have 17p loss. Clearly, there are prospective studies going on now, phase 3, to examine the introduction of these and utility of these agents as frontline therapies rather than just simply in the relapse setting. We've been fortunate enough, as you know, to be involved in some of the biomarker analyses as part of these studies. In this case, though, now, for the ibrutinib, and I'm going to jump over to here, the reason is that there have been various reports now at meetings about the patients who develop resistance. What are the alterations that they're showing? Some of them are pretty straightforward. The patients are acquiring a mutation in the gene that the agent is targeting against. Makes sense. This is interesting because it's in the same pathway. So that would mean then that whatever genes we look at and perhaps build a resistance panel, we need to think of pathways, not just individual genes. But what was interesting to us is that two of the markers that nobody else offers or assesses in their comprehensive analysis and risk stratification toolkit for CLL are actually coming up now. And this is what's detected on our mapper, deletion of 8p and gain of 3q. And on top of that, our research studies have very narrowly pointed down as to where those sites are. So that's becoming a very interesting research question now. We are now actually involved with certain collaborators to do similar types of studies for the idelalisib. Those studies are ongoing right now for patients who develop resistance, and it's the subject of one of our abstracts submitted for ASH. The other thing I want to talk about, just briefly on this slide, is how the clinicians are talking about, all right, these two drugs, while they are targeted therapies, you do see side effects. So now they're talking about if I put a patient, and some of the side effects include colitis and some other things that would make the patients miserable. So the disease is doing all right, but the side effects would prevent them. So now they're talking about doing trials and examining how can we switch patients between these two. And we've been involved in starting to work with some of our collaborators on this, as well as they're starting to ask, can we also integrate some of the other new agents on the list, such as the AbbVie ABT199, which is the BCL2 inhibitor. Can we bring that in to help as well? So different targeted. What are the genetic mechanisms involved? What are the cells that we're targeting? Which ones are remaining if the patient is resistant? And we're very happy to be involved in those research studies. So I'm going to go to the other main disease that I'd like to talk about today that we've made progress in, diffuse large B-cell lymphoma. Totally different disease. It's aggressive. Patients, for the most part, are actually treated up front. Again, median age of diagnosis is older, and it represents about 30% of all non-Hodgkin's lymphoma. Unlike follicular lymphoma, which is considered incurable, DLBCL has been potentially curable with immunochemotherapy, Rituxan with CHOP, where today about 60% of patients are cured. What's interesting about that is that there's a very clear timeline here of how well you can assess the response of the therapy. We know that by 2 years, if most patients are going to relapse, they do that within 2 years. So for this disease, the major clinical challenges involve these. 1, who's most likely to relapse within those 2 years? 2, who's most likely to have a poor overall outcome? And of course, now, how can we identify those patients who are most likely to benefit from the new therapies, which includes ibrutinib and idelalisib? Our current components of DLBCL involves FISH to look for some of the poor markers here, rearrangement of the MYC locus, our cell of origin subtyping, GCB versus non-GCB, which is becoming very important in this era of the new therapies. We know that some of the subtype, the non-GCB, appears to respond better to some of the B-cell signaling pathway inhibitors than the GCB pathway. We also have to look for immunohistochemical, look at some of these genes such as MYC, BCL2 to look for the so-called double hit DLBCLs, which have a particularly poor outcome. We had very limited TP53, MACBA, of course, with our copy number, which I'll go into now, and we have a new panel for DLBCL, which I'll mention at the end, that's in the development and validation phase. We believe that this program is going to have improved clinical utility by further definition of the role of the copy number variations, including and be able to integrate them with the mutation panel that we've developed. And most importantly for this disease is to look at the landscape of these alterations that you see in patients. For example, do all the patients who have a MYC rearrangement also carry all the poor mutations? We need to know that. So I'd like to go into a little bit of information for you about how we've progressed with our copy number validation work and discovery work. So in a large study that we did using bioinformatic tools, we took 3 independent data sets of DLBCLs. And I believe that it's over 400 cases total. And we looked to see across them what are the genomic copy number alterations that are consistent and observed within 2 groups, or 2 that overlap. And we found that there are a total of 32, 17 gains, 15 losses. But what happened is these were the broad. And we knew from some prior work that we actually have different important sites in some of the larger ones. So by a process, again, of bioinformatic analysis, we were actually able to narrow those down more and actually obtain 50 sites that were minimally commonly altered regions and with peaks. And in fact, these 50 sites carry at their peaks about 112 genes. I've listed over here you can't read it, I know that, the 32 sites that are gained and lost in the 3 different data sets that we used to use for the discovery project. I want to tell you now something about these 50 sites and the genes and how the correlative work that we're doing with them. First of all, the genes and the peaks. So what I've shown you here is some of the genes that are at the peaks. And I'd just like to walk you through a couple of points that we should recognize from this. One is that it provided us with proof of principle. Some of the genes that were at the peaks were genes that were already known and important and found in DLBCL. So we had confidence then. What was also important is that you'll see I've listed at some of these the number of genes. And that's actually very important when you're doing these genome-wide surveys. You don't want 300 genes at one site because you're going to have to follow up on each of these. And that was very important for us because it actually targeted us in on some of the genes that are involved like JAK2 that we know were strict targets, some of which, for example, the CD58 and the beta-2 microglobulin are involved in immunosurveillance and disruption of it that occurs in lymphomas. It's also allowed us to target in and find individual genes like this one here, C02, and here, that actually become brand new novel genes that are involved in this disease, and as part of their pathway, they represent novel therapeutic agents, targets. What is also important on this slide is the fact that there are a number of genes listed here for which they're involved in pathways for which there are new therapeutic agents out there. So if you, in that sense, then you may be able to use this assay and use these genes and peaks in order to be able to better classify your patients as they go into the trial, and your agent then may have a better response given the abnormalities that they show at the genes of target that are being targeted. What else are we doing with this? We're correlating it with gene expression. We have 5 significant pathways, including 2 that if we had not have found, we would have been disturbed about. We have an additional 3. They are therapeutic target pathways. We've associated with outcome, and all of these others are being done with, again, other independent data sets. We have markers of outcome in at least 2 of 4 independent data sets. And the last item that we've addressed in large numbers of samples now is the issue of genomic complexity. In this disease, if the genome is complex in this particular assay and type, it is very clearly associated with poor outcome. This also now is the subject of an ASH abstract submitted for ASH for presentation. So again, we realized that copy number variation is not the only thing that's going on in the genome of this particular cancer. We're also going to have to look at the mutations. So to that end, we've been developing a panel and designing a panel for NGS. In all of these, they're being designed so that they don't necessarily have to be done on the fresh tissue. They can be done on paraffin-embedded material as well. And in this case, we've designed it such that it's almost 200 genes, but it's very focally focused on these 3 diseases here, in particular DLBCL and follicular lymphoma. The panel has been designed to contain actionable targets for some FDA-approved drugs. It's also been designed to take into account clinical trial targets. It's also been designed to take into some that have known clinical relevance, such as B-cell homing as well. And that's part of that progresses to the next one, where we relate them in biological pathway. Because as these different targeted agents come in, they're targeting a pathway, not just simply a gene. And that's important so that we assess the pathways. We've also included some because of their frequency and cross-study validation. We've done this so that it could be used both in a clinical setting as well as in a biomarker analysis for pharma trials of these drugs that we see are coming up. So it's important for us now to be able to look at not only at some of the copy numbers, the sequencing, we have to look at cell of origin. So you'll see now that I'll show, and I believe it's the next slide, actually it's the following slide. Some of our collaborations that we now do is involved in being able to do this comprehensive testing and to look at the landscape of these alterations and features in the disease as a whole. So for some of our poster presentations, I've listed here at ASH and US CAP, oral presentations in the last year or so. We have one coming up in Turkey, and also our submitted abstracts this year for ASH. And to finish is the slide of our collaborations that we have. These are current ones that are ongoing now. One addressing a very important subtype of DLBCL that's in the central nervous system, and now our 2 large studies which have been announced recently so that we can look at all these biomarkers in the same set These data sets are very well clinically annotated, so we have very good outcome. And what's interesting about the second one, as Panna pointed out too, is that we have a single county hospital from which all these patients are being seen and treated in. So thank you for your attention.
So as you can see from Jane's work, it's very, not only are we going deeper and deeper into the hematologic side, but we're also very much focused on making sure our content is unique and can be used both in trials as well as in routine patient care. So we'll open up for some Q&A.
Jane, I was wondering for the next-gen sequencing-based panels, you're combining all these subtypes into one panel, and is that correct?
The CLL is separate,
Oh,
7 genes.
Okay.
That one is on its own.
But for...
That's a DLBCL, mantle, and follicular.
Those are,
Yep.
yes. The reason for combining, sorry.
Yeah, so I was going to ask, in terms of from a clinical trial standpoint, that makes sense, but do you think clinical utility or from payer standpoint, would they be interested in paying for an entire panel every time you're testing for a subtype of the patient, or?
So we have discussed separating out the mantle cell lymphoma for clinical utility because it would reach a different type of disease. The DLBCL and follicular are very biologically related. And the reason for that is because you heard Dr. Farrugia talking about follicular lymphoma and it transforming. It actually transforms into DLBCL. So that's the reason for linking those two together.
I'm just wondering, with these increasingly focused NGS panels, are you seeing increased interest from biopharma, and also how much interest have you seen on these potentially new druggable targets?
That's a great question. The answer is very much yes. That's, I would say, last month, that's consumed most of Jane and I's time has been the biopharma. And so, yeah, it's. Go ahead.
It's repository of DNA is waiting to be tested.
Yeah, we're actively doing that. Thank you, Jane. Very good. We have Dr. Howard McLeod. I know we were supposed to take a break, but we can't. So we're going to move right into the next section. And Howard McLeod, just some background. He's at Moffitt Cancer Center. He's a kind of globally renowned expert in pharmacogenomics. He was on the board and the scientific advisory board here at Gentris, and has a lot of scientific relationships and collaborations. And we asked Howard to join our scientific advisory board post-merger, and he has a lot of insight into really how pharmacogenomics and oncology are coming together to really now individualize medicine further. And so I'll turn it over to Howard for the next 10, 15 minutes to give some perspective and then open it up to Q&A. Howard? You want to
well, thanks, Panna, and many thanks to you and Scott and Jane and the rest of your team for the interactions we've had so far. I apologize to anyone who was looking forward to the break instead of me, but if you just go outside the room, the bathrooms are right there, so it's not too bad. I think I remember my way around there. I want to talk a little bit about the state of personalized medicine in terms of what's going on both clinically and in drug development. Hit a little bit on the specifics for oncology, although I realize that there's certainly excitement across a lot of different therapeutic areas. And then lastly, mention a little bit about some of the collaborations that are going on between CGI with both the legacy Gentris team as well as some of the newer work with the CGI team between Moffitt Cancer Center and that group. Just as a point of background, many of you won't know that Moffitt Cancer Center has quietly become the third largest cancer center in the nation, just behind MD Anderson and Memorial Sloan Kettering. Although the way the numbers are looking, we may end up passing one of those this year. Bigger is not necessarily better, but we have a lot of patients that need help. We're the only NCI designated cancer center south of Atlanta, and the top ranked cancer center by U.S. News and World Report in the southeast. So it ends up being a lot of patients come here, and so that's why the volume is so large. The personalized medicine really, over the last 10 years at Moffitt has been something that started off as really more biobanking and making sure that there was high quality tissue preserved and associated with good clinical data. And then now that has expanded into much more of changes in patient care, and not just in the tucking away of tissue for safekeeping at some later date. Now, the state of personalized medicine generally has really been driven by a couple of different factors. One has been on the regulatory side, and certainly the FDA has put out guidance that has made it pretty clear that they expect biobanking to be part of clinical trial development, that they expect analysis to be part of that as well, to understand adverse drug reactions as well as differences in dosing or efficacy. And then they've also expected that they can figure out post approval some of the untoward events that will be appearing as drugs start getting tested in millions of people instead of thousands of people. And so certainly the regulatory, I wouldn't call it pressure, the regulatory encouragement has grown over the last few years. The pharma companies, certainly the larger pharma companies, have not only done this because the FDA suggests it, but have started to see some real value in terms of having objective data to kill a development pipeline early, having objective data to fractionate markets so they have a higher chance of getting activity in late clinical development and therefore more rapid approval, and then also focus the marketing efforts so that they can have a greater value, expect a higher reimbursement rate because of the greater value in a particular patient subset. And we see that most in oncology because the data is stronger there, where a drug might be priced at $20,000 a month because there's such enrichment of benefit, whereas in other segments it would be a fraction of that in terms of pricing. The efficacy side also has come with that. We now have drugs that might be only active in 5% of lung cancer patients, but have a very significant degree of activity, not typically curing a patient with metastatic disease, but certainly providing them with a 6 to 8 month extension in quality lifespan compared to what would have been seen previously. And so oncology now, we're seeing a huge growth in personalized medicine as part of routine care, not just as part of clinical trial development or other aspects of that. We'll come back to that point briefly. That is also being seen across other segments of medicine. Certainly within cardiology, there's a lot of use now of pharmacogenomic markers, but also genetic predictors for in long QT syndrome, other sudden death syndromes, heart failure syndromes, and really trying to make it so that patients are more of the cause of disease is identified and therefore can be acted on. And it's doing things like influencing how frequently echoes are obtained in the cardiac patient, all the way through to changing which therapy someone might administer in that cases. We're seeing a bit of an increase in the influence of genetics on surgical management, looking at patients that might have an inherited syndrome as opposed to sporadic disease, both in terms of cancer, but also in terms of cardiac syndromes, rheumatologic syndromes, other areas. And so identifying patients who need elective surgeries to prevent downstream events of some sort is starting to be something that we're not only seeing more of clinically, but insurance companies are supporting that effort because of the downstream costs of care. Now, in terms of the drug development specifically, I've mentioned quite a bit of what's going on in that area, but I think the big changes is really in the much more systematic use of personalized medicine during early drug development. And so we're seeing clinical trials. It used to be that phase 1 was around the toxicology and dosing. Phase 2 was defining activity in a specific disease type, and phase 3 was a comparison of that drug or that drug combination with the standard of care, kind of
can we knock the king off the throne and become the new king type of a model in phase 3. Now we're seeing a lot more activity in what I guess you would call phase 0.5, where you will either do a clinical trial in a subset of patients, almost more like a registry approach, getting a large enough number of patients to try to better define who it is that's responding, or look in, and if you have a molecular marker to begin with, going in and enriching for that patient population initially to see whether there's enhanced activity. In many cases, the evidence of activity is just to justify the now expenditure of tens of millions of dollars to do the rest of the phase 1-2 pipeline, as opposed to using it to now enrich specifically for that patient population. So what I mean by that is you might have a group of patients with a particular disease that respond best if they're an extensive metabolizer of a drug, but you then go in and so you do that study, you show that those patients are having as good or better response to a medication compared to your competitor, and maybe the drug is already approved. You then go into classical drug development, not with the enriched genetic strategy, but rather with looking at all comers, but with the confidence that there is at least a major subgroup of the population that will have an extremely good response to therapy. And so even in companies that don't have a marketing department that has the confidence to segment the market, you can still use this sort of personalized approach to understand who's going to be, is there at least a group that's going to respond, justify the expenditure for doing the classical development approach. Within phase 4, we're also seeing a lot more surveillance going on and a lot more biobanking going on, and then putting those two together. And so there certainly have been drugs that have either been pulled off the market and then because of genetic information brought back on the market, or drugs that were not pulled off the market because it was identified that people with an abnormal bilirubin glucuronidation syndrome were those who were having the hepatic signal, and therefore one can justify that. One doesn't need to go in and take the drug off the market because there's a clear reason for that situation. So in the situations where there's no biobank sample and no molecular data analyzed, one just has to pull the drug off the market because there's no way of justifying and doing anything else. And so that sort of effort is now paying off in very specific cases for some of these companies. And then just to get back to the specifics of oncology, really trodden over most of that ground, but certainly at our place, I think at most major cancer centers, certainly at UNC and Duke there in the Triangle, there's a large number of patients that are now having molecular analysis because they've run out of objective clinical options. So you go through the therapies that have been shown to be active in clinical trial, you run out of those options, the patient is still fit and wants more therapy, you then do molecular analysis of the tumor to guide that therapy. We're also, at least in our situation, also doing germline analysis, because if you're going to do a risk-benefit discussion, you need to have objective data on both risk and benefit. And so somatic data from the tumor sequencing gives you some data around benefit, but it's the germline where the risk is happening. You're getting lung toxicity or nerve toxicity or gastrointestinal tract toxicity. You need to have data that predicts for those tissues, not just the tumor. And so we're more often now combining tumor sequencing with germline normal tissue sequencing so that we can go in and have a discussion with a patient about risk and benefit, not just one in terms of that. And I think the days of thinking of yourself as think of yourself as a company, but also thinking of patients as a tumor or a germline, we really need to bring both together in order to make the best decisions for patients. And I think that affects the way we do drug development, but also affects the way we do patient care. And frankly, that was one of the exciting things about working with CGI is that with the different mergers and acquisitions, there's now a company that can really think about both and not just a germline company or a tumor company, et cetera. And I know Gentris certainly has been in both spaces, but now with the merged companies, one can really think about how do we look at this leukemia or this lymphoma or this colon cancer in a more definitive way, and how do we look at this patient and their ability to handle the drug in a more definitive way. And that combination can be really powerful going forward. Just to finish off, I want to mention a little bit about some of the collaborations that we're doing. There's certainly the legacy collaborations with Gentris keep going strong, and Scott Clark and many of the others that are probably in the room there that we've worked with. We have a number of publications that are already out and more coming from past collaborations when I was up at the University of North Carolina, and that is now expanding as we've set up shop down here at Miffitt Cancer Center. And so these studies can be in areas like which antiemetic, which anti-vomiting medicine should we be using to make sure our patients can receive adequate care without extraordinary side effects, through to which pain control should we be choosing, expanding out into a number of other areas in terms of which drugs should we choose for the treatment of cancers. We and certainly Scott and I have been working through a number of those projects that are going forward, including some that are active right now. So it's a past tense and a present tense in terms of the work that's going on with the legacy Gentris company and the Moffitt collaborations. And I know Gentris has these kind of collaborations with a number of the leading cancer centers, not just with Moffitt. The CGI discussions are now taking place with the folks leading hematologic malignancies, so leukemia, lymphoma, myelodysplastic syndrome, as well as we have a very active and internationally active HPV and viral oncology program. And the HPV program in particular, with the CGI having such an active role there with both research and clinical management, we're now having those discussions about how do we have the company and these key opinion leaders do some really active research and active practice work that can enhance the value of what CGI is doing, but also enhance our ability to manage patients. And so there will be some of that is research, but research using clinical grade CGI products, and then there will some of that will be we'll just be looking to take on as part of clinical management, not so much as research. So I think the collaborations with Moffitt are focused around more around the therapeutics currently, but also will expand out in terms of prognosis and in terms of patient management, even things like who should be vaccinated in a more intensive way, or who should be doing something different on the viral oncology side. So I think the last 10 minutes have been all over the place in terms of the landscape of personalized medicine, certainly what we're doing in drug development, or what is being done in drug development and in oncology in particular, and then a little bit about some of the collaborations with CGI. So it's probably been something for everyone and nothing for everyone, but hopefully you got a little bit of a feel for what we're doing, and I'll stop at that point and open it up for questions or whatever Panna wants to do.
Dr. McLeod, you mentioned that a lot more people are thinking about both germline and somatic analysis. Do you see anyone else out there analyzing both, aside from the CGI Gentris acquisition?
Well, I think so right now they're in the oncology space. There's not a lot of direct competition, and so I think there's some real market share opportunities there. There are some very active folks in the tumor space. Foundation Medicine being one of those companies that they could easily add, or not easily, but they could try to add germline analysis, but just haven't gone that direction. The folks, the folks that have been doing germline molecular testing, the large ones, but since your LabCorp's backyard, the LabCorps, certainly could try to add those types of things onto their portfolio. The difficulty, both from a drug development and from a clinical aspect, is that it's not so much the commoditized genomic analysis where the value is. It's the specialty analysis and the interpretation and support that really is needed both on the drug development and the clinical side of things. And so, that's where some of the large places have kind of fallen down, and where the smaller, more focused, customer service-oriented places are starting to excel. And so that's why I'm a bit bullish about CGI, is that opportunity not only has it been active in both companies separately, but now putting them together, I think that there's some real opportunity to go forward. Other companies could come into this space. Obviously, they won't have the long track records, but currently we have trouble coming up with who the competitors are going to be in terms of that, at least with the companies that are out there currently.
One more question before we take a break.
So just so I understand this a little better, let's say on the commercial side of things, so are you guys eventually going to be, per se, like for example, maybe offering a NGS panel for CLL, and then in addition to that same panel, it also has a germline on top of that, so it kind of just hits everything all at once?
We're architecting the offering now. I don't know if it would be on the same panel, but it would be the same offering. Yeah, so someone for CLL, they can get both the information about the risk stratification and therapeutic option, and the same sample also get the pharmacogenomic profile of the patient. Go ahead.
So as Dr. Farrugia mentioned earlier, most of the panels that we make, it's very easy to add. Yeah. And that addition will also depend on the number of polymorphisms that we see, the germline, that are associated with whatever the feature is we're looking for. If it's 5, 3, depends on what it is. But they're very easy to add.
So we'll take about a 15, 20-minute break. Thank you. It's a very uniquely positioned product. I have Dr. Marie McLeod on the line. Marie, are you there?
Hi, good afternoon, everybody.
So she will be providing color commentary along with our field product manager, Anna Marie, and they'll be walking through FHACT. And then we hopefully will have lunch by the time they're done, and then we'll have an expert, Dr. Hopley, who's one of the leading OBGYN guys in and also in cervical cancer, and he'll be calling in and talking about how he uses it. He's probably one of the biggest power users today of the FHACT product, and so we've asked him to dial and give his perspective, why he uses it, where it's going. And so we'll have we'll go through the FHACT product, maybe have lunch by then, and then have Hopley call in. So Anna Marie, please take it away.
Thank you. Well, good afternoon. My name is Marie Samulis, and I am the field product manager for FHACT. And I think after this presentation, you will certainly understand how our test will take the guesswork out of cervical cancer screening. Believe it or not, there still is, even in the United States, some issues with the current cancer screening programs that I think we will be able to identify and illustrate to you, and you can see how succinctly the FHACT product will fit in very nicely. Now, FHACT in itself, the name is FISH-based HPV-associated cancer test. FISH is the methodology that we have chosen to use because of its ability to be reproduced as well as being very sensitive and specific. Now, cervical cancer screening worldwide is still a very big issue. And the fundamental reason of that is because in those nations there is not a concise screening program that is readily available. So more often than not, once the disease is found, it is found at a very late stage. In the industrialized countries, like the United States, we have enjoyed the PAP screening program, which was instituted in the 1940s and has been hugely successful and certainly saved the lives of many women. However, that in itself is still not exactly where we as a society, with all our knowledge and genomic ingenuity today, need and could be. So today, I'd like to take you through, very simply, how cervical cancer screening is worked up in the United States. So it starts out with a woman who attends to her physician. More than likely, it's an OBGYN, who then procures a sample of a PAP, and it is placed in a liquid-based cytology, which is vastly used today. This liquid-based cytology specimen is sent off to a PAP laboratory, a pathology laboratory, so to speak, whereas then they analyze the sample, and if that sample comes back normal with a diagnosis, that woman just goes on for routine follow-up, frequently every 3 years, sometimes every 5 years, depending. However, if an abnormal diagnosis is made, such as L-CIL or H-CIL, or even more importantly, inconclusive diagnoses of Ascus agus or Ask rule out H, then HPV testing enters into the picture. Why? Because HPV is the causative agent for cervical cancer. Now, by itself, it's just a virus. But something happens in some women, whereas that HPV infection will integrate into her genome and cause cell replication, turning the switch on for cervical disease. So if a woman who has her HPV tested is positive, then she's sent on to colposcopy and biopsy. If it's negative, then she's really in good shape and she can go into a routine follow-up. Now, there's some issues with what's going on today in the PAP world. Well, first of all, with the liquid cytology, it's not perfect, okay? It has imperfect sensitivity. Too many false negatives, meaning that there's truly disease, but it's not being identified. The results can be uncertain and unclear, such as these right here. This US at the end of these really denotes of unspecified significance. They know something's going on, but they're not really sure what it is. So when HPV, a good HPV test came around in the 1980s, it really shed a lot of light on the whole scenario, because HPV being the causative agent, you really want to know what your HPV status is as a woman. If it's negative, that's great. If it's positive, you are now at risk for cervical disease. So today's HPV testing, the various numbers that are out there, it doesn't distinguish between a transient versus a persistent infection. As I said, HPV, similar to the PAP that shows too many false negatives, HPV shows too many false positives. It's important to understand that most HPV infections that women have never result in a cytological change. But those women who fall into this category here are then sent on for colposcopy and biopsy. Colposcopy, biopsy, invasive procedure, of which if it's a biopsy that's procured, they're taking pieces of that woman's cervix. Now that in itself is diagnostic for sure, but it also can be detrimental to that woman's cervix insofar as that she may then have problems getting pregnant, infertility, as well as problems maintaining her pregnancy. Back in 2010, this topic of cervical cancer screening was really talked about in CAP Today, which is a publication for the College of American Pathologists. And these national thought leaders came to a consensus that really said, unfortunately, knowledge gained from the existing tools, what do we have? PAP, colposcopy, biopsy, cannot predict which lesions will progress to cancer. Dr. Dara feels that we need something more specific than just the presence of the virus. Remember, the virus is the virus, but unless and if and when it integrates in, it means nothing. Dr. Wilbur from Mass General basically indicated that we need new markers that can show this transformation. We need to be able to distinguish those women who have the potential of perhaps going on to cervical disease versus those women who are not at risk. And Dr. Ungar states that she really likes the triage potential of 3Q. So what FHACT was designed to do was really to fulfill this unmet need. What the goal is to identify women at the earliest stage possible as to who in this big group right here, the vast majority who our immune system clears viruses all the time, 90% of this group will definitely either regress or some of them will just stay in a quasi-infection but not go further. No integration has occurred. A small percentage of these women, of which today in the United States, this is about 2.5 million women, have abnormal testing results. 10% show that they will go on, if left untreated, into more significant disease. So our solution at Cancer Genetics is FHACT. There's no resampling. Basically, the same vial that is taken from the woman at the time of her PAP is tested at the PAP laboratory. HPV testing is also can be performed. If that vial warrants further testing, then that vial comes to Cancer Genetics so that then we can provide this very valuable information of genomic assessment. So what are we looking at? Well, it's long been found back in, I think it was in the 1980s when 3Q was found to be significant in the cervical cancer arena. So our assay is actually looking at 4 chromosomes with independent loci. 3q26, 5p15, centromere 7, and 20q13. And all of these, what we're looking for is gain. Are there additional numerations of this area? 3q26 by itself basically has been observed in about 75% of cervical carcinomas. Our study, which we added the CEN7, 5p, and 20q, allowed us to increase this detection up to 89.5%. So what does this mean? Well, this is clearly illustrated in this case study. There's really 2 points of the FHACT test. Number one is the true power of this test is in the negative. Because if you have 2.5 million women who have abnormal test results, they're then subclassified. And if the vast majority will clear their infection, wouldn't you want to know that as soon as possible? Because today, those 2.5 million women are going on for further testing. So as a negative, this patient here, she was diagnosed with LSIL and a positive HPV. Remember, as I said, 90% will not go on, 10% has a potential to go on, of which all of these people are now, women are going on for colposcopy. Does the patient present any markers of high-grade lesions? Well, how would you know? You run the FHACT test. So this particular patient, all were negative, 3q, 5p, 20q, and CEN7. We look at over 2,000 cells, which is very significant in itself to make sure that we are capturing as much activity as possible. What this clearly shows is that there is no chromosomal instability in this patient, and certainly there was no sampling required. Now, how might a clinician use this information? Well, because no integration has occurred, the clinician may decide not to refer that woman for immediate colposcopy and may choose to just place her under active surveillance. And they should take FHACT into consideration with the whole context picture of the patient. What is her history? As I said initially, that globally, it's still a huge problem, cervical cancer. And by being able to move this test forward into the market, we can have the opportunity to really touch a number of lives. And how are we going to do that? Well, we're going to create a footprint for our FHACT test in a number of steps. Step one was, is we already know that there is a need in the market. We have myself, field product manager, who is out talking to a various number of entities out there. Identifying target OBGYN and pathology groups. Understand the partnership with CGI is to the pathology group because they perform the Pap smear. The OBGYNs order the PAPs, therefore they order the FHACT test. Much information is on our website at CGIFHACT.com, and we had our first published article in June of 2013 with NCI. Step 2, which we are in the process of doing, is the introduction of FHACT to targets. Webinar series, which we have routinely. Connecting with KOLs. We have a number of potential targets in place right now, of which we are in quite a number of discussions. Consolidating the data for additional collaborations, and then, of course, look at the economic side. In step 3, certainly VIP visits to our laboratory, establish and track the database and the outcomes, submission of trial, support of regional data and global trials, leverage early adopters. There are a number of clinicians out there who are very eager to utilize this type of testing. And then the 4th step is continue our quality improvement, additional work with any regulatory bodies that we pursue, and localization of regional needs. Inclusion in the guidelines. This is a very significant step that we certainly believe that we can accomplish. And we look at ourselves as a potential partner in cervical cancer screening. We're not looking to replace HPV, we augment HPV testing. As far as moving forward in the regulatory world, supporting FHACT in early adoption, currently, as has been a foundation of CGI, is there are patents in place. The test is currently offered as a LDT, laboratory-developed assay, in our CLIA CAP-certified laboratory in New Jersey. We recently received CE marking, which then allows us to offer this test abroad. We're looking to have the patent issuances by the end of 2014. And these 2 goals right here, I think, are very significant because much of what is done today is driven by the ASCCP ACOG guidelines. And there are a number of people in this arena who realize that you need a triage test, because you may be aware that FDA has given approval to Roche, and in the near future, you'll be hearing a lot about how HPV testing will be moving as the primary screen in women's health. Well, again, HPV testing will determine what your risk may be, if there is any. You still need something further down the line, that genomic transformation, and then FDA approval. So our operational focus, certainly which continues on a daily basis, is in the production side. Currently, the PRO's manufactured and outsourced in India, and this will be relocated to CGI India. The test analysis, looking at greater than 2,000, and many times that's what we do look at, over 2,000 cells, it's a very lengthy process. So we are in the process of validating an automated platform. With this comes the ability to actually remotely analyze the samples. Reimbursement is very important, of course, and we know what a changing landscape that is. Today, the test is currently being coded as a FISH probe test. We are initiating contracts with third parties and talking to these individuals as reimbursement based on a value base. Because obviously, all those extra tests that are performed, whether it's the office visit, the colposcopy, the biopsy, there is an additional cost. And many of these women, you need to understand in the screening algorithms, they are returning to their clinicians every 6 months because they need to resolve what's going on, and it's very unclear that this occurs. So it's very costly to the society overall. Our goals certainly are to continue to comply with the new LDT regulations which have been released, our patent issuance, further validation with NCI, which is in play right now for 300 plus samples, We're looking to gain 5 major pathology partners, of which we have preliminary conversations currently with a few. Additional publications, this is certainly very important and will continue to do so because the data will tell the story and the data will show that FHACT is a test that needs to be in the triage for women's health. Establishing national health economic studies, cost-benefit analysis that needs to be done. And then evaluating the FDA approval process. Potentially, this test also, because it is HPV related, it could be utilized for other squamous cell carcinomas, which anal and head and neck.
All right, so I believe you guys would like me to talk about the FHACT test and how it may be useful for clinicians and pathologists alike. I'm just going to talk about it and see what benefits we've already observed. I'm unsure as to the level of understanding of my audience. If anybody has any questions, if I use any terms or concepts that you're not immediately familiar with, please don't hesitate to interrupt. So, I guess I'd like to start by saying that until recently, I think there's been an unmet need in cervical cancer screening. Cervical cancer screening has been tremendously effective in the U.S. It has led to a significant decrease in the incidence of cervical cancer. The one problem with the Pap test, or Pap smear as it used to be known, is its lack of sensitivity. This was never usually a problem historically because the cervical lesions are so slow in developing that if a lesion was missed on a Pap test in the first year, when the lady would come back the second, third, or hopefully by the fourth year, these lesions would be picked up. When HPV testing was added to cervical cancer screening after the human papillomavirus was described as being the causative agent behind cervical cancer, this significantly led to an increase in sensitivity of the Pap test. The problem with HPV testing is that while it does increase the sensitivity of the Pap test, is lacking in specificity. As you all may know, about 80% of women will be infected with HPV over their lifetime, and the vast majority of them develop no cervical disease at all. And even those that do, the cervical precancers may regress as the body's immune system clears it of the HPV. So one problem that we see is women with minor or no cytologic abnormalities that test positive for HPV. This HPV may be persistent. They may have persistent low-grade cervical disease, and they're constantly being biopsied and literally picked at by their OB-GYNs. And this can be very stressful, painful, expensive. And a lot of these women are walking around, they feel like they're a time bomb. They're infected with this virus that causes cancer, and they just don't have peace of mind. And for whatever reason, their body is taking its time in clearing this infection, and this is the unmet need that I'm talking about. How do we stratify these patients? How do we treat these patients or reassure these patients? And I think the answer lies in this new fish-based DNA damage testing, as we see with FHACT. When we have a negative FHACT test, a negative DNA damage test in these patients, this can widely reassure these patients that the DNA damage that causes or leads to an increased likelihood of cervical disease or cervical cancer is not yet present. Similarly, when these tests are positive, this is a red flag for the clinician and can say that these patients do need follow-up. They do need more biopsies or more aggressive biopsies or surgical intervention. Any questions on this so far? No? Okay, we're good. So that is it in a nutshell. We have already seen doctors starting to adopt this new technology. We have a lot of patients with atypical results on their Pap tests that are positive for HPV. Their doctors would traditionally biopsy. They have started to integrate the biopsy results, the Pap results, and the FHACT test results into the treatment of these patients. And the vast majority, well, I'd say all of our clinicians that we deal with are very, very happy with this additional piece of information, because it really is an additional, very important piece of information that was previously lacking with these patients, and what to do with them. Some clinicians have tried genotyping, looking for more. These are strains of HPV that are more common in cervical cancers. That is one way of stratifying these patients, but there are disadvantages to that, and that are not met as with the FHACT test, that the FHACT test does address. So we've also seen patients who have had minimally abnormal cytology results that have had a positive DNA damage test that have subsequently been further investigated by their clinicians, and they've been found to have high-grade disease in there. So that's the FHACT test has benefited these patients enormously. So again, that's where I stand on this test. Any questions or comments? Okay, that's an excellent question. I'm going to repeat it because you're very faint, and I want to make sure that I understood and everyone else got that. Have the clinicians started to integrate this into their screening guidelines, essentially? Is that correct? Yes. As of yet, it's not been adopted into the official guidelines that many of our clinicians follow, the ASCCP or ACOG. Those guidelines are often way behind the current technologies. Relatively recently only adapted to involve HPV. So, however, saying that, some of these clinicians have set up individual protocols with us. For example, if a patient does have an atypical cytology result and is HPV positive, then they would like the test done. So sort of a reflex testing, if you will. Some people would like it on any patient who is HPV positive. And then a lot of our clinicians tend to like it on a case-by-case basis. If a patient is a young lady, she has a high-grade PAP or a high-grade biopsy, and they want to avoid doing a rather aggressive excision on this patient because, for example, she wants to preserve her fertility, then if the patient and the clinician would like peace of mind, can they watch and wait this lesion for 6 months to a year? They may do the test and incorporate it that way on a case-by-case basis. The point being, if this was a negative test, the clinician would feel safer watching the patient for a year or so. So I hope that answered your question.
Yeah, that was great. Thank you, Dr. Hopley. Any other questions for Dr. Hopley? Hold on.
Dr. Hopley, was wondering, so if you could clarify, I think you talked about it a little bit, but in terms of different clinicians adopting the test, what level of education with regards to this test or other types of testing currently exist? Meaning, I'm just curious as to, do you go out there and is this part of your marketing, sales and marketing effort?
Absolutely, it is in several ways. Our sales representatives will go out and they'll give literature on this testing as a whole, and then on our, the specific testing that based upon the FHACT test. A lot of our clinicians are educated in that they do follow current literature, and this has been in the literature in many forms relatively recently. This will be something that we educate our own clinicians on when they call asking for advice on how to treat, how to manage a particularly tough patient. So as pathologists from doctor to doctor, when we're talking to the OB-GYNs, we will talk to them. It's actually something I'm planning on writing. We have a quarterly newsletter when we provide our clinicians opportunities for education on new tests or new concepts, basically information we'd like to get across to the clinician that we might not have the ability to in the normal course of affairs. And that I actually write this newsletter, and that's actually going to be a topic for our upcoming newsletter. So absolutely, yeah, we try to have as much communication as possible with our clinicians. And it's something that they're very receptive to. Our docs are very, very busy. they're packed, they're seeing and managing patients and performing surgeries and answering calls and emails. It's literally from dusk till dawn. And so they appreciate any contact that they can have with us and any way that we can keep them up to date with what tests are out there.
And then my follow-up is, for the doctors who are not adopting the test right away after they've been, they have been educated about the test, what's the biggest barrier to adoption? Is that just they want more clinical data, or I'm just kind of curious as to what currently the doctors who kind of decide not to do it?
I lost you after biggest barrier to adoption, but I can tell you that I think a lot of our docs like to follow the guidelines, and this not being in the official guidelines as of yet, that may be a barrier. I think that this is only a matter of time. I'm sure as the literature grows and these tests become more widely adopted and standardized, then this will just lead to increased familiarity with these tests. And as soon as the guidelines reflect it, then I'm sure that will be the last barrier to adoption, yes.
So my question is, are there any other companies out there that are working on a similar test as FHACT that you're aware of? And if you're not, is anybody at Cancer Genetics aware of such a test?
Are there any other companies out there working on similar FISH-based cervical cancer tests? Is that the question?
Yes.
Yes, I'm terribly sorry. Yes, there are, I can think of at least, 3 out there. The FHACT test is unique in that it offers, in my opinion, greater specificity because it has a greater number of probes that look for more diverse and more numerous abnormal chromosomal gains. For example, I know that Quest has a test that just looks for gain of 3q, which is perhaps the most common gain, an abnormality that one will see in cervical cancer. There are a couple of other companies, I think, that may offer more than 3q, but not. The FHACT test is unique in that it has 4 probes which look for gains in 4 chromosomes. So, in my opinion, this will be lead to a more specific test.
Well, just clarification, 50-50 in-house versus sending out to you guys. Any reason for that? Is it just the capacity issue? Or because if you're able to keep all the samples in-house by doing it in-house, and do you have plans to expand that for doing the in-house testing further to, could close to almost all of it, or just kind of curious?
So my answer is, we're trying to market test ways to get the product broadly distributed. We think this is a 2 million test market for us. Okay, so could we easily internalize 2 million tests? I don't think I'd be 100% truthful if I said the yes to that question. So this is part of a distribution strategy, going with a leader in gynecological oncology like Path Advantage and Dr. Hopley. Can we use this to more broadly distribute it? This will help us think about and drive future decisions. So Dr. Hopley, the question is why do some of the samples are processed at Path Advantage and why do you send some to us?
Oh, okay. No, that's a great question. Sorry, we have just literally started offering this in-house, and as we work through any technical difficulties, which we're actually experiencing very few, we just wanted to keep the load light for our operating technician, so she's not overwhelmed as we start to offer this to our clinicians. This is going to be a short-term, just a bridge to when we're performing everything in-house, which will happen very, very shortly.
Banu will give an overview of the renal portfolio and what we're doing in kidney cancer. And this is one of the most exciting areas. We've done a lot of work. We've got very, very compelling panels. And then we will take a quick break after this and then do the tour of the Gentris facility. So, Banu.
Good afternoon, everybody. I'm Marie Gowrishankar. I'm working for CGI from 2010. and I'm a principal scientist for the urogenital program, as Panna pointed. Prior to coming here, I was working with Fox Cancer Center, where I was looking at the genetics, epigenetics of renal cancer. So this area is not new to me, so it's really interesting. And the first slide shows you the market cap. As you can see from here, almost 3 to 5% of adult cancers is renal cancer, and about 13,000 people are dying every year because of this disease. And there are currently 42 clinical trials ongoing for various purposes. And the unmet needs for the cancer right now is to classify various subtypes in the cancer as such, and also to predict metastasis and for therapy selection, which I'll go over in a few minutes. So if you look at renal cancer, the outcome mainly depends on the stage of the cancer as such. Stage 1 is mainly the tumor, where the tumor size is less than 7 centimeters. Stage 2 is little bigger, but still confined within kidney. And stage 3 is when the tumor starts to spread to the neighboring veins, but still it's confined to the kidney capsule. Stage 1, 2, and 3 are called locally invasive because and they do not spread to distant organs. But stage 4 is the one which is dangerous. That's when the cancer starts to metastasize to different organs. It can go to lung, brain, or bone, or pancreas. Lung and bone are the very common sites of metastasis for renal cancer. The therapy also depends on what's the stage. As you can see, the best response is for stage 1 and 2 patients. When the cancer starts to spread, that's when the overall survival becomes very, very less. While stage 1, 2, and 3, they just require either just a small portion of the tumor cut out, which is partial nephrectomy, or the entire kidney, depending on how big the tumor is, to be cut out. But stage 4 people, they require additional therapy along with nephrectomy. Kidney cancer is unique in the sense that most of the chemotherapy do not work with these because the cancer is refractory to chemotherapy. So the only promising agents are systemic therapy, targeted therapy, in other words. There are totally 7 approved drugs for the disease till now that FDA has approved, and among which a majority of patients are treated with the first drug, which is sunitinib. So although all these drugs are existing, currently they are given in a trial and error basis, and there is not real predictive biomarkers so far to actually say that these patients are going to respond to this drug. So with this, CGI has developed 3 main products. One is an array CGH based platform, which is Urogenra, or other words urogenital array. And we are also coming up with a more robust sequencing panel, which is our proprietary NGS panel. I will tell in the details of each of these in a few moments. And lastly, we are also giving a complete approach wherein we are integrating multiple platforms and in order to give the clinicians the overall picture of what the patient's cancer is about, so that will help the clinicians better guide and treat these patients. So there are 3 main unmet needs in kidney cancer. The first thing is the diagnosis. When the patient walks in, he either has a bigger renal mass, which is more than 4 centimeter, or small renal masses. For bigger renal mass, it's pretty straightforward. The patients they undergo nephrectomy, and the surgical specimen is big enough that the clinicians can distinguish the subtypes clearly, except for a few percentage of unclassified cases. So these are the 3 main subtypes on which kidney cancer is mostly about. There are about 5% of patients which are unclassified, and those are problematic, and they require genetic profiling to actually guide treatment. But the real problem comes with small renal masses, because these masses are generally taken by needle core biopsies, whereas everyone pointed out the tissue is so limited that a clear H&E staining and a clear classification is not possible by, I would not say not possible, but it's just that it's challenging. About 20 to 30% of them are non-diagnostic so far. So this is where we could offer a molecular test wherein we can clearly distinguish benign from malignant. Why we need to distinguish? Because the treatment is different. benign cases do not require nephrectomy. Nephrectomy, is first of all, removing kidney itself related to a lot of comorbidities and unnecessary cost and everything. And mostly kidney cancer comes when the patient is around 50 to 60 years, and you don't want to undergo unnecessary surgery if it's not necessary. So the second need is prognosis and prediction. Here we are trying to identify the genomics of metastasis. As I told you before, metastasis is the main, it's the marker for, that's the one that guides the outcome. If people who have metastasis, they have very poor outcome. So we are trying to understand what are the genomic changes that happens in a metastatic patient. The overall idea is to see if these markers can serve as predictive biomarkers in the early stage patient to say that so and so patients are going to metastasize in future and we can treat them differently. And the third unmet need is the theranosis, where, as I told you before, Sutent, although majority of people are given Sutent at this time, only 50% of the patients show complete response. So here we are trying to understand what genomic markers are present in the patient to actually say that these people are going to respond to a particular drug, which is sunitinib in this case. So regarding the first diagnostic challenge, what we did was we took over 600 genomic profiles that were available in publicly available databases. And then we also had in-house studies with over 100 neoplasms, both benign and malignant. We did array CGH profile on them. We devised an algorithm which we call through the Eurogenra assay. And the algorithm actually, it has 16 genomic regions. Based on just the presence or absence of these 16 genomic markers, we will be able to clearly classify them as benign or malignant, and also clearly subtype them more specifically, whether it's clear cell, papillary, or chromophobe, or other subtypes. So after we designed the algorithm, what we did was we went on to validate in collaboration with Cleveland Clinic. We had over 190 formalin-fixed paraffin-embedded specimens. We extracted DNA and did array CGH on Eurogenra for these samples. We were able to get a result for every single sample. We got 100% diagnostic yield, and the sensitivity was pretty good for distinguishing between benign and malignant. And even for distinguishing between the various malignant subtypes, it's 93% sensitivity. We have obtained we have filed patent for the algorithm currently, and we have obtained patent for the previous, for the Eurogenra as such. The real validation for this particular, for Eurogenra is needle biopsies, because that's where the clinicians are actually facing the challenge of clearly subtyping them. So for this, we went ahead and collaborated with Memorial Sloan Kettering Center with interventional radiologists, Dr. Coleman and Solomon. And we took over 50 needle biopsies. We did a blind study here. We ran all these, every single biopsy on the Eurogenra, and we classified according to our algorithm. And then we compared it with what the clinicians would classify them as. In fact, we were able to classify some unclassifiable or non-diagnostic specimens by the clinicians. We were able to give a proper classification. An example of a case report is shown here, wherein this patient went on to get a needle biopsy pathology. As I told you before, about 30% of the needle biopsies are non-diagnostics. So in these cases, what the clinicians do is they ask for repeat biopsies, or they ask them to undergo surgery. So in this case, that's what happened. The patient went on to get the surgery, and upon surgical removal, they found that it's a clear cell subtype. Without knowing what the subtype was, we got the same needle biopsy specimen, we extracted DNA, we ran them on Eurogenra, we got, this is the genomic gains and losses that we found in that sample, and according to our algorithm, the same sample would be a clear cell subtype. So, which gave us more confidence that we could work with these needle biopsy specimens, and we could help the clinicians in such cases where the biopsy classification is challenging. We are currently expanding the clinical utility of the algorithm. There are several benign neoplasms for which there is not as much study available in the literature. In fact, we are conducting a large genomic characterization study with over 70 benign renal neoplasms to understand their genetic profile. An interesting challenge here is that although these are all benign neoplasms, what happens is a small subset of these benign renal neoplasms, few years down the lane, they start progressing to metastasis. So we are trying to understand the genomics right now to see, can we stratify, can we actually identify those aggressive benign ones and help the clinicians. The other collaboration that we are doing with Westchester Medical Center is to genomically profile hemangioblastomas. These are there is a diagnostic challenge in this field because it is often confused with metastatic clear cell, which also goes to spine. And hemangioblastomas are spinal tumors. So with this, I'll go on to the second unmet need in kidney cancer, wherein the CGI goals are to predict markers for metastasis. Currently, there are some risk stratification models that are available. MSKCC is a pioneer in doing this kind of risk stratification models. Currently, the patients are roughly stratified into two major groups, good or intermediate and poor. Good or intermediate are given sunitinib, sorafenib, and interferons as first-line therapy, whereas poor risk people are given temsirolimus. So since these risk criteria are existing, but they are based on the clinical features such as hemoglobin level, LDH level, and so on, they are not good enough, robust enough to actually select patients for therapy. As I told you, that not all patients respond to these therapy. So they will see if a patient develops resistance, they'll go ahead and give the next therapy. So the clinical need here is to try and incorporate more parameters into this risk stratification models and see if we can better stratify these patients. So we are working on that right now. And the other interesting thing is the therapy nowadays, there is very good therapy for the depending on the site of metastasis. For example, people who go on to bone metastasis are very poor prognostic markers, and they have found that cabozantinib works very well with bone metastasis. And for brain metastasis, they are treated with targeted radiotherapy. So it's important to know where the tumor is going to go. Depending on that, the treatment also, appropriate treatment can be given to the patients. So to do this, what we did was in collaboration with Dr. Mozer, who is a pioneer in leading some of the clinical trials with sunitinib. We obtained over 130 clear cell specimens. We actually stuck to clear cell for these things because clear cell is the most predominant subtype, and that's the most malignant as well. And so over 130 samples were genomically profiled. We used whole genome arrays to find out what are the genomic markers here. We identified 29 genomic markers for metastasis, and we went on to validate in two large independent data sets. We used TCGA as one of our data sets for validation. Out of this 29, we were able to validate 7 biomarkers in both these independent data sets, which was good. We can actually go ahead and now test these 7 biomarkers to see are they real predictors of metastasis in independent data sets. That's what we are planning to do now. So the other thing that we did for site-specific markers is we again profiled some metastatic lesions. We found 9 markers that were associated with lung metastasis and 3 were associated with bone. And we were able to validate it in an independent data set wherein we were able to identify 4 of the 9 markers were in fact validated in an independent data set. So which gives us confidence now. We are going to now test it in another independent data set in a retrospective study. So we have presented most of our classification as well as our metastasis work in several conferences and meetings. And in fact, we have also published a portion of our classification study, and we are in the process of publishing the remaining work as well. There's a lot of R&D efforts going on, and because we are validating with several independent institutes that we would like to publish them, and because these are what we are doing is really novel in terms of the genomics research as well in kidney cancer. So I'm going to change topic here. What we are doing now is, so far what I told you now is so far on Eurogendra's clinical utility, wherein copy number changes are mainly assessed by Eurogendra. But as you know, cancer has all kinds of changes. that now we are involved in, we have actually designed a proprietary sequencing panel wherein we can also screen for mutations in these patients. In fact, in the last couple of years, there are, although kidney cancer is a quiet genome with respect to mutations, but now due to this deep sequencing approach, there are many studies that are coming out now, and some target mutations are becoming important in prognosis as well. So we want to explore that area as well. And our sequencing panel includes a lot of SNPs which are involved in prognosis as well as diagnosis. And we also have, based on our in-house studies, we have some clues on genes which may be involved in sunitinib response. And we have designed this panel with all these markers. And now we are going to do validation on both fresh tissues as well as FFPE-based subtypes and needle biopsies. So that's what we plan to do. And lastly, since cancer also has protein changes along with mutations, it also has rearrangements. So we are offering a complete testing wherein we could check the immunohistochemical levels of the proteins, as well as we could check the copy number information. We can check the gene rearrangement as well as mutations to give a more complete picture to the clinicians as to this. This is actually one step further to help clinicians do personalized medicine. That's it, and I'm open to questions. Thank you.
I was just wondering if there's anyone else that's out there predicting the metastasis site. Maybe I'm just naive, but I haven't seen that.
No, actually, for NGS panel, there's only one other company there, which is actually which has a panel for looking for hereditary mutations in kidney cancer. Other than that, I have not come across. In fact, I checked even outside US if there is any other company that has, these kind of tests. Nobody has.
As a follow-up to that, I guess, are you, when you're trying to determine when a patient is going to respond to a certain drug, are you looking at the mutations or the genes of the cancer cells, or typically of the body's healthy cells and how they metabolize certain things?
No, we actually take, all these studies, I didn't really elaborate how these studies are performed. For the sunitinib response, what we do is we take the tumors of the patients who are given sunitinib and whose follow-up data is also available. So, but those samples are taken before the sunitinib treatment. So we try to look for markers in the tumor samples to see whether the so-and-so patient would respond or not. Because we have all kinds of patients here. When we take 100 samples, let's say 60 people would have responded and 40 people would not have responded. What we do is we divide them into two groups and see, can we distinguish based on their, genetic changes into two separate groups.
And then when you're looking for where the metastases is going to go, are you trying to predict where it's already gone or where it will go in the future?
Where it will go in future. That's the overall goal. So to start off with, we are looking for we have identified markers that is important for a particular that will predict where the cancer would spread to. Then, of course, all these things should be validated in a real-time setting in a low stage tumors to see whether they are true predictive markers or not. So that's something we are just going to start now.
One more question that came from your presentation, just about mutation in cancer in general. Is it that most cancers, or do we have an idea of cancers are caused by mutations that occur environmentally or over time? Or are these mutations present in the body when we're babies or young adults that will eventually progress to the disease? And then just because you have a certain mutation, it doesn't necessarily tell you that you're progressing to that disease maybe. So what other markers should we be looking for to tell us that information? And can DNA give us that?
So let's address it from the beginning. I'm sorry. Let's address it from the beginning. So a germline polymorphism or alteration or mutation have been observed in certain diseases that will predispose you to certain types of cancer. And there are many examples of that. what is it? Is another disease where we know that there's mutations in VHL, and those people tend to get kidney cancer.
That's hereditary.
So that is hereditary. So that's germline. Now, do certain cancer types share types of mutations? Is it related to environmental exposure? Yes. So now it's coming up, if you look out in the literature, there are certain types of squamous cell-like lung cancer, and I believe also a subtype of bladder cancer that are often related in the fact of their mutational spectrum that you see. And the reason could be because they're all in tissues that are exposed to environmental agents. There are others, though, that are more unique and tissue-specific, like breast basal, carcinoma. Renal cancer is a group that's totally on its own. It doesn't share many with any of the other diseases. So bladder cancer is going to turn out to be a totally mixed bag, some that share the environmental and some that don't. Now, in terms of, what was the last one? Oh, in terms of the types of mutations that you see, there are clearly mutations that we call drivers. They're highly significant in driving the tumor or the transformed cell forward. But you also need the acquisition of additional mutations to result in progression of the disease. So there are two different levels. There's a third level, which we would call passenger mutations, which they've occurred, but we don't think they're actually contributing anything functionally. And that's important to tease out those from the others that really have functional roles.
So do you need additional clinical data to show that the complete package has clinical utility over ordering just the Eurogenra, for example, by itself? I'm just curious as to when you're marketing or when you're taking it to, to the clinic, like how do your customers sometimes order the complete versus in a more a la carte fashion?
Yeah, that's a good question. Why, part of it is philosophical, why complete, and then the validation is a separate one. So if you go to our complete, go to renal complete as a backdrop. A lot of the, as you know, the space is moving rapidly, and people still rely on existing technologies, whether they be immunohistochemistry or FISH or IHC. These are existing technologies that are out there, immunohistochemistry, FISH, et cetera. And people still want that information. And part of the strategy of complete is that we want the sample regardless. And so if someone is used to using FISH, so for example, CCND1, or someone's used to using certain IHC, and they want that information, they shouldn't have to go send that information to Quest or LabCorp and then send us the Eurogenra after that, because they're not going to do it. So part of the strategy of complete is to make it simple or easy for that person to send the sample and get all the testing at our site, bundled with our proprietary content. Is it more valuable? Probably, because you get a fuller snapshot. Also, in some of the more challenging or complex diseases, like CLL could be an example, or DLBCL, there are a lot of different information that you get, whether it be from IGHV rearrangement or other types of testing. And each piece gives you a piece of information. Different clinicians, as you know, might feel more comfortable looking at IHC, and some might feel more comfortable looking at FISH. And so we don't want to have any kind of burden associated with us going to market. So complete allows us to basically market and do all the testing for that site. And a lot of people want to get our proprietary test, but they want to see how it compares with the existing technologies that are out there. And typically a complete for us is existing types of testing brought together, and then our proprietary product built into that. And they can compare and contrast, or they can use everything, or that way they can make an integrated opinion. And so that's why we market the complete. Otherwise, the challenge you often see, and for those analysts follow areas like cancer of unknown primary or other areas, the IHC is done somewhere else, and the cancer of unknown primary is done later, 6 months, 3 months, and oftentimes the sample doesn't go to that lab. There are issues retrieving the sample, getting the block. There's issues with comparing what they had in the IHC versus what the cup array told them. And so it's just an impediment. It becomes a challenge. And so we want to remove that challenge as much as possible and give a more complete snapshot, because sometimes these other technologies give information. And so you want to make sure that they continue to get that information.
Hello, I'm Scott Clark. I'm the Vice President of Global Scientific Operations. I have a Ph.D. in cell and molecular biology and did my postdoc at the University of North Carolina in conjunction with the EPA. So that's what moved me down here and decided the weather was so nice I'd stay here. I've been in contract labs and with Gentris for 13 years now, starting in an initial company with our founder, Michael Murphy, and then have stayed on throughout Gentris, and with the acquisition by CGI, continue to stay on as the global head of the science operations. So I currently oversee the genotyping, sample management, nucleic acid purification, scientific development, and the China operations. So I see most of all the scientific operations. And so what you'll see as I go through the lab tour with you is certain parts of where a DNA sample comes in or a blood sample comes in, and what happens when that arrives to how it gets to the patient and the doctor, and how they can do some treatment on the patient, especially in the clinical trial arena.